All right. Good afternoon. I'll just share my screen. So I'll be presenting our project called the Velvet Worm Slime, a natural, fully recyclable biopolymer. My name is Prof. Ali Miserez, and this work is done by research fellow in my lab, Lu Yang, Ken, and Bharji Sharma. So what is this project about? We've been interested in an interesting animal called the velvet worm. And this animal is actually fairly old. It's actually a phylum by itself. It's carnivorous. It's a terrestrial invertebrate. The reason we're interested in this project is that it actually secretes a very interesting biopolymer, which turns out to be fully recyclable, only made of protein. And we believe that this has the inspiration for a future green biopolymer. So let me just show you a worm that we have in my lab and what it does when he feels threatened or he feels there's a prey. See, he will fastly eject the slime. The slime then will harden very fast and make a very strong fiber within a few seconds, as you can see. Let me show you again the video. And as I'll show you in a few seconds, what is really intriguing with this slime is that this biopolymer is that if you put it back in water, after a while, it will completely redissolve. So this is basically a fully recyclable biopolymer, only made of mostly amino acids, that is protein. Okay, so when we started this project, there's a few things that are known about this slime. And so, of course, the slime is initially liquid in its gland before it secretes. It formed this adhesive slime that is ejected under shear force. I just showed that to you. It's highly extensible. It has a fairly high tensile strength while it's cured, and you've seen that it cures after a few seconds. And of course, what is interesting for us from a bio-inspired perspective is that it's fully recyclable. And the composition is mostly protein, lipid, some sugar, and some water. So there was work done 10 years ago on trying to understand the slime protein. So this is a electrophoresis gel showing the protein here that have been extracted. And there is some partial sequence that have been identified in this large protein, but it's only partial. I insist on this. So people don't know the full sequence before we started this project. Those proteins are highly charged, and there is some hypothesis that they might assemble through electrostatic interaction. If you take the slime and you resolubilize, and I'll show that again in a second, you form those small nanoparticles, and those nanoparticles will then assemble into fibers under shear. So this is the high molecular weight protein. It's disordered. There is some phosphorylation of those proteins has been detected, and there's also the hypothesis that there's electrostatic interaction that self-assemble into those globules. But those are some questions that were unknown until we started that project. There's only partial high molecular weight protein that's been sequenced. There's only prediction of phosphorylation. It has never been identified. And as I mentioned, there's only a partial sequence. The protein structure and slime formation, there was some work that was recently conducted, and it was found through X-ray diffraction and Raman spectroscopy that there is beta sheet inside those nanoglobules. Then in the fibrils, there's also through X-ray diffraction, it was identified that there's some beta sheet that are about 20 strands, sorry. And then, as mentioned earlier, there's some charged and hydrophobic region in the protein sequence. And so the current model that we are actually challenging now with our data is that there is beta crystalline stacking that is possibly enhanced by ionic interaction, which is mediated by phosphorylation of phosphorylate amino acid. So this is the model that people have come up with recently, where they think that there is those slime droplet that you see here, those spheres, and as the slime is being ejected, it is subjected to strain force, shear strain forces, and those shear will induce beta sheet formation. Then you have water being removed and fiber formation. And the intriguing thing, as I mentioned, is that if you put back in water, it will redissolve into those monodispersed nanodroplets. So if we want to mimic this slime material, there's a few questions that need to be solved. First of all, what is the full sequence of slime? That's absolutely critical. And at the moment, or before we started that project, there was no full-length sequence. The structure of the slime, what are the phosphorylation site on the protein? That's actually very important to understand. And maybe they could have some other post-translation modification and also other protein involved. So the velvet worm, actually, the actual worm name, scientific name is called Onychophora. We actually have been working with 3 species. This one is the family of Peripatopsidae. And actually, you find it in New Zealand. And then the slime is obtained from our collaborator. Those worms here, we have them live in the lab. I show you one. We actually, one of them is a species that we found here in Singapore forest. It turns out it could be a new species. So we have now a little colony. And another one is actually from Costa Rica, but we have a distributor from Malaysia who sold us a few animals. And both of them now, they have produced little babies. So we have a whole colony. And so in the big picture, what we're doing in this project is the following. We're doing mRNA sequencing, so next generation sequencing of the gland where the protein is being secreted. And so we've constructed the whole transcriptome library that we've sequenced. In parallel from the slime, we're doing high throughput proteomics, including identification of phosphosite. And then from that, we've identified the full-length protein or proteins, because there's more than one that we've identified. We identify which one is the most abundant. And we also have found the key post-translational modification. And we now, thanks to those data, we're able to make a hypothesis on fiber formation, which we believe is more accurate. And in parallel, we're also working on the characterization of the crude slime. So the crude slime, we're doing an MR, TM, AFM to really try to draw a sequence property relationship. And now that we've actually identified the full-length protein, we are expressing it in E. coli or part of the protein. And then we are aiming to identify the self-assembly mechanism from the recombinant version. And so those are the main topics that we've been pursuing on that project. In case you're interested in how we keep the worm, so as I mentioned, we had one of the species from the local species we found in Singapore in the secondary forest. It was a lot of hard work from Ken Lu Yang from my lab, and he finally managed to get one. And we were very happy because, as I said, they have offspring now. So we have about 10 of each species. And in order to do NMR, we actually have an interesting strategy that we have cricket, live cricket that we feed with addition of carbon-13 labeled glucose. And then those crickets are being consumed by the worm, and that allows us to enrich the slime for NMR analysis, and that has greatly helped us to get very precise structural analysis. So in summary, in this project, we've identified a protein sequence, a full-length protein sequence in slime of those 2 species of velvet worm. They're obtained, they're verified, and we have the high molecular weight protein. We also find alternating patches of positively and negatively charged amino acid, but there's some difference, and there's potential to form localized beta sheet structure. We've done some prediction of the structure based on some modern bioinformatic tools, especially AlphaFold that was in the news recently. We actually, interestingly, did not find phosphorylation in the main high molecular weight protein, but we have some other post-translational modification that we think are very important. And one of the key results is that the high molecular weight region of the slime protein is actually made of 2 protein complexes that are connected by very specific intermolecular bonds. And that plays most likely a very important role in fiber formation. And with this, I'm done. The funding, of course, is through by ExxonMobil through the Singapore Energy Center. And I want to thank also, again, Adam Uzadi and Giovanni Piloni for supporting this project and for having great scientific discussion since we started it. It's been really, really great. Thank you.